From Microbe TV, this is Immune, episode number 39, recorded on November 30th, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back.
It
is disgusting, rainy, and yucky here.
It's raining
today.
here too. Isn't it supposed to be snow this time of year?
It has been incredibly warm. We've had flurries once. That's about it. It's very unusual for this time of year. We were outside, walking around, doing yard work, no coats.
Yeah, I did something yesterday that will guarantee it doesn't snow. I moved my snowblower into the garage. It's not going to snow now.
My car is currently at the shop getting its snow tires on, so it won't snow.
Also joining us from Durham, North Carolina, Steph Langle.
Hey there. Good to be back. I think we were with each other just last week.
Yeah.
So 2 times in 2 weeks.
Is that too much?
No. It's great to see your faces, hear your voice.
And from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. It is equally gross outside here. And I think that means that it's a good day to stay inside and do a podcast and then go to the lab.
There you go.
You have a long drive to the lab, don't you?
It's very long. I mean, given that I can see it out the window right now, it's real tough.
I didn't know that. That's cool.
Yeah, I dream of such a thing. But
oh, yeah, your commute is.
As a grad student, I lived right down the street from Mount Sinai. It was great. But I slowly got further and further. Mm-hmm. Today, we thought we'd give you an all-email episode because we haven't done email in a while, and we should catch up. And there's some good questions, many good questions. Yeah. We don't want you to think that we're ignoring you.
Right. So I know some of them are dated back from the summer. So it actually will be fun to answer those because we will have perspective from the last, 4 to 6 months as the science has progressed, and it has progressed quickly.
Yes, it certainly has.
Mm-hmm.
All right, so let's go do some of these. I'm going to have Cindy take that first one.
Give me the long one, right?
Well, some of them are really long. This is not the longest.
All right. Nah, this isn't the longest one, but they're all so very interesting. So it says, Dear Professor Racaniello and colleagues, since some time I've been listening to your immunology podcast, loved the episode on checkpoint immunotherapy and CAR T. And since the COVID-19 crisis, I've been listening regularly to This Week in Virology as well. Have recommended the podcast to many friends and colleagues. Since some time, I've heard about people who suffer a long course of COVID-19 disease with chronic fatigue syndrome like plain. A colleague of mine manifests these symptoms, which really sucks. And a friend who is an occupational physician has told me about many patients with similar complaints as well. Recently, Ed Young, a great journalist writing for The Atlantic, wrote about this phenomenon. Ed Young compares the symptoms of this chronic COVID-19 course to myalgic encephalomyelitis, chronic fatigue syndrome, or ME/CFS. And he puts in a quote from the article, "It's not clear why this happens. Akiko Iwasaki, an immunologist at Yale, offers 3 possibilities. Long haulers might still harbor infectious virus in some reservoir organ, which is missed by tests that use nasal swabs. Or persistent fragments of viral genes, though not infectious, may still be triggering a violent immune overreaction, as if you're reacting to a ghost of a virus," Iwasaki says. More likely, the virus is gone, but the immune system, having been provoked by it, is stuck in a lingering overactive state. I was wondering your thoughts on this. Best regards, Gerber. So I found a couple of interesting things. One of the things I found was, if people are interested, there is a recent reflection by a physician who is one of these long haulers and describes it. And what I thought, and this is in the Journal of American Medical Association, what I thought was really interesting about that is he talks about that he's changed his perspective on patient empathy. And the idea that before he would disregard many of the, annoying little symptoms that a patient had and dismiss them. And now that he's kind of suffering this and he's, stuck in his basement for like 60 days with intermittent fevers and aches and so forth, he's like, I have a totally different respect now for patients when they're saying they have these, symptoms that were just kind of like, that doesn't fit anything. That can't be, it's all in your head type thing, which was interesting. But generally, though, I don't know what you guys have to say about these long haulers and your thoughts, but I think, Akiko's on point. I think there's multiple explanations and we don't really have a full answer yet. What do you guys think?
Well, I know that the TWiV team did an excellent episode called Long-Term COVID and ME/CFS. It's an extension of Vincent's relationship with, can you remind me of his name from Berkeley?
David Tuller.
David Tuller, who has been really active in the ME/CFS community and trying to combat this false narrative about these patients that it's just either them being lazy, they need to be reconditioned, it's about them, being able to get up, but in fact that they actually regress of a lot of patients. And so that episode, check it out. In regards to my feelings of this, I think I wish people could just from the start empathize with others so you don't have to have this story in JAMA where you're like, I wasn't empathetic and I never understood, but then I got the disease and now I'm empathetic. If we just start from the beginning, think about how many more patients, we could actually reach and treat and take seriously. In regards to the science, I find the incidence of multi-inflammatory syndrome with COVID to be really interesting. We're seeing it in children and we're seeing it in adults. I think it was back in 2005. There was a paper published by Jeff Kuhn, I think it's C-A-H-N. He's at UTMB now. He was at Yale. And it was the association of one of the common cold coronas with Kawasaki's disease. Kawasaki's disease is another of these maybe virally induced inflammatory syndromes. We're seeing it more in women, which has a relationship maybe with autoantibodies. And so I think the signal of these type of diseases with viral infections is not strong enough unless you have a pandemic. We don't see these things happening in other circumstances because it's like hit or miss. You see a patient with it, you treat them, they get better or not. And they just get lost. And I think COVID is really amplifying that this is serious and these people are not making it up.
Yeah, I agree completely with everything that Steph had to say. So we talked to 3 different people on the TWiV episode about long-term COVID. In fact, one of the things we talked about was what the preferred terminology was. And they debated a bit, but tended to go with long-term COVID. And one of the things that came up on that episode was the idea of this being something about autoantibodies, and there potentially being some autoantibodies in a number of these syndromes, but this being a situation where we're only now getting enough of a signal with the pandemic. And I've been kind of intrigued by that idea of those autoantibodies ever since then. I originally would have said something similar, though less eloquent than Akiko Iwasaki in that quote.
You're as eloquent.
Well, thank you.
Credit.
But I think that I'm now starting to wonder about that overreactive immune system and how it might relate to autoantibodies. I believe it was also that episode where they mentioned that by and large, a lot of physicians had considered something that was a medically unexplained syndrome as being sort of not a thing, or the symptoms must not be real if it was medically unexplained. And that really got me with the idea that I was shocked that we thought that we could explain everything, because clearly there's a lot we have left to learn. And so I think that this is highlighting how much we have to learn, and hopefully some of these COVID-19 patients' conditions might give us some discoveries that'll help people with some of the other conditions as well.
It makes you wonder how medical schools train physicians if they're not empathetic, right? And I think that they're lacking some training. And I know that medical schools have this in their curriculum, but it must not be a priority because so many non-empathetic physicians go out there.
I actually think people misconstrue empathy and sympathy. And I think that there's a lot probably a lot taught on,
Yeah.
be compassionate and sympathetic.
Yes, but not
Versus
understanding.
empathy is just stepping in that person's shoes and acknowledging that even if you disagree with them or even if you don't believe them, that what they're experiencing is actually real. And so I think that that's a little bit harder. And so, yeah, I think you're right.
I mean,
they think they're teaching empathy, but they probably aren't.
The nice thing about that TWiV 680 Gerben, I definitely recommend you listen to it. We had Dave Tuller, who has been fighting this MECFS writing for years. He's been fighting this PACE trial, which says if you simply make these people exercise and give them psychotherapy, they'll get better. And this is just, the trial was a farce and it's not true. And now we have a similar syndrome with COVID-19 patients. We had two people on the pod, Maddie Hornig, who is a professor at Columbia, and she works on MECFS.
She's a physician.
She's a physician, and she developed COVID-19 and now has long-term COVID. And then Fiona Lowenstein is a science writer, TV producer, who also had long-term COVID, and they both spoke of their experiences with them. So I highly recommend that. It's really eye-opening. We were actually going to have Ed Young on that episode, but he backed out at the last minute, unfortunately. But because he wrote this Atlantic article that Gerben is referencing. So there's a lot out there, but we don't know what's going on. I don't think there's any virus involved, frankly. I think the theory is going towards it being something else, as Brienne said, maybe autoantibodies, but it's not going to be one thing. It's going to be multiple causes. Very interesting.
It's very interesting. I agree, Vincent, that it's likely not the virus sticking around. I mean, there are things like defective viral particles that we don't really fully understand, but likely your body clears the virus, but it's an inability to regulate some type of inflammatory response that is not going to be the same between person to person. I think there's going to be a genetic element. So, but it's hard to study because we need animal models, and these are complicated.
Exactly. Yes. There are no
syndromes. We were trying to, Sally and I were trying to write a grant to study MISC, so the multi-inflammatory syndrome in children in rhesus macaques. But there's no model for Kawasaki syndrome. There's no model for this. So, to study that in an animal, I don't know. It's very hard.
One more thing I want to mention is that I've followed for years an MECFS patient, Jenny Splotila. She used to be a lawyer, and then in her 20s, she became sick and couldn't work anymore. And she's been doing a lot of activism, and she has a very nice blog. I think it's called Occupy MECFS, something like that. Anyway, she wrote a blog post. She said, I could scream. I've been telling people for years, and now all of a sudden, long-term COVID is now people believe it, and they didn't believe us. And it's, so she's... socially, right? That's interesting how different communities are interacting with each other, because on one side, it's like, finally, there's recognition, we'll get funding, we'll understand. But then it's like, hey, we've been here for a while yelling about this. So, and it is interesting historically when you think about over the course of history, if autoimmune diseases are more... landing in its sex space. So more women have them, likely because of estrogen. And the gaslighting that happens in the medical community related to women who maybe have these unexplained conditions. I think historically you can look back in time and see this is to be a very persistent trend. Yeah. That term came up many times during that episode, gaslighting.
What's that?
Gaslighting.
Oh, gaslighting. Yes. Yeah. It's a helpful term.
All right. Brienne, can you take the next one, please?
Sure. Mikhail writes, Dear Immune, greetings from Moscow. I am a longtime fan of both Immune and TWiV, and it is very useful for me as a researcher. Thanks a lot. On Immune 32, June 5th, you joked about infecting people with SARS-CoV-2 to study cross-reactive pre-existing memory activation. I think we found a way to do it without infecting anyone. As you know, T-cell receptor, TCR diversity, is extremely high, and because of this, you could use TCR sequence as a barcode to quantitatively track individual T-cell clones in time. The idea is to collect samples longitudinally, sequence the TCR repertoire on each time point, and look for TCR sequences which increase and later decrease their frequencies after the infection or vaccination. Works great with the live yellow fever vaccine. 2 donors we sampled and sequenced TCR repertoires last year for a different project got mild COVID-19 this March. So we were able to sample them again after infection and use TCR repertoire sequencing to identify T-cell clonal expansions and track them back to the pre-infection and pre-pandemic T-cell repertoire. Surprisingly, a bunch of COVID-19 reactive CD4 clones, but few CD8s, were found in the central memory subpopulation from 1 year before, suggesting cross-reactive memory indeed participates in immune responses to SARS-CoV-2. Here is our preprint. First author is Anastasia Minervina, also a big fan of yours, describing these results. Thank you again for your hard work, Mikhail. And he is in the Department of Genomics and Adaptive Immunity at an Institute of Bioinorganic Chemistry that I have not heard of in Moscow and probably would butcher.
You should, wait, let me see. You should try it. Look, it looks fun.
Shemayakin Ovchinnikov.
It's not bad.
That's not our Russian
Bioorganic chemistry, not inorganic, just, small difference.
A bit.
So this is an interesting, it's on BioArchive, but it was posted in May. Let's take a look. Something must have happened since then.
I feel like I've heard of this story, so I'm wondering if it's been published since.
They would usually say that on the BioArchive page, right? It's published.
It does say it. I mean, I haven't pulled it up, but it would say that, yes.
Yeah, it doesn't say that it's been published yet. It's probably under torturous review.
Mm-hmm. Yeah, I think the scrutiny on these types of studies where they're looking at TCR repertoire and sequencing them it's becoming more strict as more papers have come out. So I'm assuming it's languishing.
I think the bottom line here is that they see memory CD4, right, and CD8 in these patients. Is that correct? And they can characterize what the T-cell repertoire is.
What do you think about that, Brianne?
I think I'd have to look at that paper a bit more, because I have a hard time completely determining how they would identify that cell as a memory cell,
right?
Particularly as a central memory cell, as he mentions here. But I'm not sure if I'm familiar enough with the assay to totally address that.
They say in both donors, the majority of infection reactive clonotypes acquire memory phenotypes. And certain CD4 clones were detected in the memory fraction at pre-infection, suggesting participation of pre-existing cross-reactive memory T cells. So these are themes that we've really talked about already on TWiV and Immune. So quite interesting.
Right. And just looking briefly at the paper, I see there are some principal components analysis plots that I know we talked about a little bit when we talked about the John Wary paper earlier. I think Cindy explained really nicely.
Steph, can you take the next one?
Sure. Yeah, actually, I'm going to hop back to that previous one. Just to comment on this concept of pre-existing T-cell cross-reactivity and what that means. I think that there are a lot of papers that are definitively showing that there are cross-reactive T-cells at maybe low levels that exist in patients pre-SARS-CoV-2 infection. I think that this is not uncommon. This has been identified in previous papers and previous patients. For instance, I had just pulled one up where they had found cross-reactivity to HIV after an influenza infection. And so I think what this means is that what T cells can bind to, depending on the sequence of the antigen, they could have similar sequences that or similar enough that would allow a T cell receptor to partially bind, but the binding is what matters. So I think when we think of functionality, we can see sequences that look similar in TCR receptors. But what does that mean for functionality? It's going to involve avidity. It's going to involve how many of these antigen epitopes are binding to the paratope on the T cell receptor and how that actually interacts with each other. So there's been 2 papers, interestingly, that have come out. We can put them up, but they found that there was a a lot more of these cross-reactive T cells than children, which signifies potentially common cold coronaviruses driving this common antigen that would increase the proportion of T cells that could respond to it. But then a very recent paper showing that the functionality of those T cells did not provide protection against severe COVID-19. So no matter what your levels of these cross-reactive T cells were, it didn't necessarily matter. And I think that's because in immunology, you're going to find low levels of things that cross-react, but they just may not bind strong enough to matter in an actual infection.
Yeah, I think I've phrased it somewhere, sometime,
in the many podcasts you've done.
Somewhere in the past, as the fact that cross-reactive doesn't equal cross-protective.
Right.
And those are two separate things.
Yeah. Great. I think, a lot more to look into, because it doesn't mean it doesn't matter, but I think we just have to read that with what somebody says down here, a Tonka truck full of salt. We will bring that up. Okay, so Kevin writes, Kevin says, thank you for all these microbe.tv podcasts. I discovered Immune back in January when I was trying to find podcasts or lectures about mucosal immunity. I just get the warm and fuzzies when I hear somebody's excited about mucosal immunity. From Immune, I discovered TWiV and then the whole world of microbe.tv and got distracted from my initial interest in mucosal immunity by all of your coverage on COVID-19. Now I listen nearly all the time and usually several times to each episode so I can absorb as much as possible. But now I've come full circle and I'm back to mucosal immunity. Vincent and others on the TWiX podcasts have noted that SARS-CoV-2 seems to replicate in the gut, but that there's no pathway for the infection to get from the gut to the lungs or the nasal cavity. And he says, fine. But is there a way for the gut infection to educate the mucosa in the lung and nasal cavity such that a stomach infection by SARS-CoV-2 would confer some immunity against infection in other areas like the lung or nasal cavity? And so he asked, does IgA, these are antibodies, T regulatory cells, et cetera, travel amongst the various mucosal sites, or are they trapped in the gut mucosa? If a child contracts the gut version through fecal oral transmission and then later contracts the respiratory version, would they have additional immunity? Also, can you recommend any mucosal immunity 101 podcasts or lectures? He's listened to Brienne's lectures, which are great, and a couple others from the Albert Einstein School of Medicine, but he's just scratched the surface. Thank you so much. Can't overstate how much I enjoy them. That's from Kevin. So a lot to touch on here. I think first we should say that there are not two different versions of SARS-CoV-2, that one infects the gut and one infects the lungs. The primary organ where this virus is replicating is the lungs and the upper respiratory tract. It is likely that you are ingesting SARS-CoV-2 that maybe winds up in the gut. We know SARS-CoV-2 does not necessarily go through your blood. There's not a strong viremia. Viremia is just the ability of the virus to go into the blood and infect maybe other parts of the body. We know that SARS-CoV-2 can infect in the gut. There's been a variety of papers that showed in really nice organoid models that they're infectable by SARS-CoV-2. I don't think it's really understood yet how replication in the gut contributes to infection. I think people find it very difficult to culture those viruses from the gut, that virus from the gut. I don't think they're like a plaque assay. They don't find any infectivity. But the question of trafficking is fascinating because there's two concepts. There is a common mucosal immune system, and what really is what defines this is the ability of B and T cells to traffic across your body to multiple different sites. I study IgA antibody secreting cells and their ability to migrate from a germinal center or a Peyer's patch to other parts of the gut, but then they will go to the mammary gland. We learned from a recent fascinating paper, they will go to the brain and protect against fungal infections. They will go to the nasal, upper respiratory. They go less to the lung. There's a lot, maybe more IgG antibody secreting cells happening there. The lung is complicated, but that in itself can allow protection at multiple sites. But at the same time, it's common, they can travel, but it's specific. It can be non-redundant in the fact that each organ has its own secretion of chemokines and cytokines that make the lung, the gut. So, you do have trafficking, but you have very specific immune responses depending on the organ.
And there have been experimental protocols where to take non-human primates, they do immunization of one mucosal and they find the antibodies at another site, right? It's very common to see that.
Yes.
Yeah.
Yes.
Mm-hmm. Yeah, go ahead, Brian. Sorry.
Oh, I was going to say that that's something that has been sort of examined a bit in the HIV field, where there's a question of whether or not you can get antibodies or immune responses in sort of the reproductive mucosa by giving a vaccine in some other mucosal site. And so there is sort of an idea or something similar to what he's talking about here. But again, the lung is a little bit unique, and I think the gut is a little bit unique too.
Yeah. And that's the concept behind mucosal-based vaccines and why they're a little bit more challenging in terms of formulation, because you have to deal with the mucosa, the microbiome. You want these cells to traffic, like if you were able to give a sublingual, like underneath the tongue, can those cells traffic to your lung to provide protection? It depends on the adjuvant and the formulation of how stable is it, because you're dealing with proteases. So it's can open up, and you're interested in mucosal immunity. Do you guys have a good, there's not like a mucosal immunity book, is there?
There is, yes.
Oh, okay.
And it just came out in a new edition this year.
Perfect. We should post that in the show notes.
We can put that link up. Yeah, it's a very good textbook, very detailed.
I want to say that I believe there is at least one SARS-CoV-2 vaccine, which is an adenovirus vaccine, put in a capsule that you swallow and it goes into your gut and reproduces there, produces mucosal immunity, and the idea will be that it protects the lung, right?
Yes.
And that's beautiful because you just, right, you don't have to inject it. And so
correct me if I'm wrong, Steph, but there's this idea that certain mucosal sites will immunize better against other mucosal sites. So this historically called, like for those of you who know the historical term is reticular endothelial system. So it's like the entire mucosal system is connected, the common mucosal immune system that you talked about. But there are some interesting things that people are finding, like what you said, where, could we do sublingual and get it everywhere? But there are certain sites that if you immunize in those sites, it doesn't protect everywhere, but it protects some of the sites. So I think there's a lot we don't understand about the address is basically like postal code, right? So some of the postal codes go everywhere. So if you educate a T cell in one place, they'll go and populate all the all the mucosal surfaces. But other ones, if you immunize in another site, they'll populate some but not others. And we don't really fully understand why that is, which makes it tricky about making these mucosal vaccines.
Yeah. And I think the zip code is dependent on, so what chemokine is that organ producing majorly that would attract the cells to come to the site? We're also in the mucosa dealing with, there are different types of epithelium depending on which mucosal site. There could, like in the vaginal tract, there are multiple epithelial layers, whereas in the gut, it's just a single epithelial layer. So there are a lot to consider. And, I love the idea of a pill-based, that company's, it's called Vaxart. And they have clinical trials of the same vaccine for influenza, and they were able to demonstrate, it was in the Lancet in January, that they saw equal amount of protection from the injectable flu zone vaccine compared to the pill-based one, equal protection in a challenge study in humans. So.
Wow,
that's really impressive.
Okay, Jesus writes, "Dear Immunites /immunos /immunes and TWiVites, how should we refer to ourselves?" This is a long email. No, it's okay. I'll go through it.
I know.
My name is Jesus. I'm a rising third-year PhD candidate at Johns Hopkins graduate program in immunology under the mentorship of Dr. Nilab Shastri. My lab studies mechanisms of immune surveillance, how the immune system can distinguish friend from foe, and cells with altered antigen processing. Specifically, I study the generation of peptides derived from non-AUG reading frames destined for MHC Class 1 presentation. I've been an avid TWiV and immune listener ever since I joined the Shastri lab 2 years ago when we were still housed at UC Berkeley. Thanks to TWiV and immune, I've learned of many papers that have shaped my PhD project and future research interests. I've grown to love virology and systems immunology. Your podcast was the only thing keeping my brain from turning into mush during the lockdown. and currently they are much needed motivation as I return to the lab and contemplate viral models for the study of non-AUG-encoded MHC1 epitopes. Johns Hopkins reopened research labs June 17th. On a side note, I want to perform a plaque assay in some way, shape, or form before I finish my doctoral studies. Vincent, how can I do this? Well, since you're in Baltimore, it's an easy train ride. Well, once we can travel again, come on up, and we'll teach you how to do it, and you can see the wall of polio while you're there.
Awesome.
Best assay ever devised. Okay, recently I presented the Grifoni 2020 study discussed in Immune 32 and TWiV 620 to my lab, and we both liked and cringed at the study. The purpose of this email is to raise a question about the methodology of this study and others like it, and share with the rest of the Immune and TWiV fan base the Shastri lens for reading papers related to immunopeptidomics. So I just remind you, this is a paper called Targets of T-Cell Responses to SARS-CoV-2 Coronavirus in Humans with COVID-19 and Unexposed Individuals. And this came from this group, Shane Crotty and Alessandro Sette and Florian Kramer at Mount Sinai, et cetera, and others. There were also people at Duke, right?
Yes.
Yes. So I think this ties in with what we were talking about before with cross-reactive T-cell responses, both whether we can detect them and whether they're protective and all of those things.
Sorry, not Duke, UNC.
Well, I was going to say, I know Duke has recently published mRNA T-cell related paper. I don't know about.
I know that paper.
Yeah, it's
so many papers.
I was looking at Aravinda de Silva, and it's
that's
UNC.
Carolina.
Yeah.
Oh, right.
Not Duke. It's, they're in the same area. It's hard to.
I say it's fine, both of them, but people get upset.
Okay.
So this is one of the conclusions is there's cross-reactivity, as Brienne said, we were just talking about. Okay, so that's the paper. Immunopeptidomics has become a promising academic marriage between immunology and bioinformatics that has great potential for unlocking the peptidome presented by MHC1 and MHC2 with recent advancements in epitope prediction and mass spectrometry, paving the way for increasingly more sensitive peptide identification. And then he or she will explain this here. And I just want to point out that when Shane Crotty comes on, he always tells us that Alessandro Sette is kind of like a master of this whole idea of using peptide pools to look at T-cell epitopes in his case. I'm sure, and others.
And I remember, oh, sorry.
No, it's okay.
Oh, I was going to say, I remember we did have a discussion when we went around about that paper, but our discussion was more about, are they really looking at memory cells? But we kind of bought it hook, line, and sinker about these peptide pools. We didn't really question that part, but this person is raising this question about these pools. And so I think it's a really interesting point as you read this.
Yeah.
Okay, so the next section is called Peptide Megapools Are Risky Business, which sounds to me like it could be a title of this episode, Or we should maybe Tonka Truck.
Tonka Truck assault. That could be
Tonka Truck full assault.
Then we get the kindergarten K through 6 crowd when we put Tonka Trucks in. Right? All right. While this method of identifying the source of immunogenic peptides is high throughput, the generation of these peptide pools and all of the peptides in this study has many caveats. The main problem is that each individual peptide in the pool was not HPLC purified, high-pressure liquid chromatography purified following synthesis. They used crude peptides, meaning that a plethora of reaction intermediates, think truncated, heterogeneous, and side chain reaction peptides and aggregates, are present in each peptide tube the authors used and eventually pooled. I guess if you did HPLC on each one, it would be prohibitively expensive, right? Because there are thousands and thousands of, they're mega pools, right?
Yeah, they were.
Crude peptide synthesis reactions can range from 50 to 70% target peptide and contain organic and inorganic salts from the synthesis reaction that can wreak havoc to cells in unthinkable ways and affect peptide binding. I guess that's a good point, right?
Yeah.
Mm-hmm. Definitely. Yeah. I was really glad that Jesus wrote in because we aren't experts in all things. So this, the generation of the peptides, they want to get the data out fast, which we can appreciate because things are happening in real time. But these are really, T cells are very responsive. They're very reactive. And if you have things in there, as order of maybe 20 to 40%, non-SARS-CoV-2 specific peptides.
This reminds me when you say we're not experts in everything. No way. And yet, that's the basis for science. You discuss things and you find people. And here on these podcasts, we get listeners who write in and explain things like this email. And this reminds me of an email which we'll do on Wednesday's TWiV, where we got something wrong. And the writer said, don't ever wander in this area again. I'm like, it's not the scientific spirit. That's how things get done. You wander into new areas and you learn, right? So,
yeah, I'm so excited that we have this email so that I can learn all of this.
Yeah, it's great.
Exactly.
All right. Even if the deprotection or addition of one amino acid to the growing peptide chain is 80 to 99% efficient, that leaves behind 1 to 19% of unreacted amino ends, protected or otherwise, that can then serve as substrates for the following amino acid addition step, generally in traditional solid phase peptide synthesis, where synthesis occurs C-terminus to N-terminus. After the generation of a 9-mer or 10-mer peptide, say for the CD8 T-cell studies in Grifoni, the final crude peptide reaction will contain anywhere from 40 to 20% or even more peptides by mass that are not your target peptide. Back of the envelope rough calculations will place the total concentration of these non-target peptides upon resuspension to be 100 to 10 micromolar, with individual off-target peptides ranging from 100 to 10 nanomolar. Now, take into account that a homogeneous population of T cells, say a reporter T-cell hybridoma or OT1 CD8s, can respond to their cognate peptide MHC when the peptide is as low as 1 femtomole. T cells are extremely sensitive, and in some instances into the sub-femtomole range. In pooling crude peptides, the relative concentration of off-target products will be higher than that of one individual desired peptide. Also, peptides can compete with one another for binding to surface MHC, since exogenous loading of peptide pulsing, as was done in Grifoni paper, to MHC relies on the affinity of a peptide for a given MHC molecule, binding groove, and displacement of a previously bound peptide. Truncated variants of a peptide can still bind MHC and, if anything, compete for access to the binding pockets. The variability in T-cell responses presented by Grifoni et al., while representing a trend, could be due to these off-target effects, especially since the authors do not indicate the severity of COVID-19 suffered by each infected individual data point, patient/donor in the study. Taking the above into account, I view the figures relating to CD8 positive T-cell responses with a Tonka truck full of salt. Let alone the CD4 positive T-cell figures where the peptides are longer and therefore introduce much more variation on the off-target reaction products. An alternative would be to elute MHC class 1 peptides from cells that are infected with SARS-CoV-2 and then running the cleaned up eluate and identifying the SARS-CoV-2 peptides by MS. No easy job by any means, requires a lot of sample by mass, and one will need patients with the same HLA alleles to remove variability in peptide sequence due to HLA binding requirements. As I read this, I'm thinking, what happened to the reviewers here? They could have said, you have to mention this. This is a limitation of this study, right? At least. Not to reject it, but just mention it. Yeah. Don't get me wrong. I enjoyed and appreciate the paper and the overall trends in antibody and pre-existing immunity it discovered. Generating an in silico list of and synthesizing peptides is no joke. And acquiring pure peptides, less than 99% pure, is an expensive and time-consuming process. The Sette and Karate labs have contributed priceless tools for the identification of peptides, and I would love nothing less than to shake the hands of each author and PI in the study. Wash your hands. Not yet. However,
later.
As immunologists interested in identifying viable targeted vaccine targets for humoral and cell-mediated immunity against SARS-CoV-2, we must be wary of prioritizing expediency for the sake of information over the careful and deliberate approach that has advanced our respective fields in the past decades. Great point. Decades. It's not even a year, folks. Not going to solve all the problems in a year. Vincent, please always keep it grumpy. Grumpy means you care. Thank you very much. Respectfully, Jesus. That's great.
Yeah, this is fantastic. It really gives us an insight into being more critical about, papers just from the perspective we didn't really know what goes into creating peptide pools for T-cell recognition.
I forgot to read the last paragraph where
Jesus
says, I appreciate every single podcast your group has published. Look forward to the possibility of hearing my question. Well, I hope you hear it. A big shout out to my lifelong friend, Teresa Lupone, for introducing me to this podcast, helping nurture my love for research. Rafael Rivera and Valerie Zapata at the MCB grad program at UC Berkeley and the Shastri Lab and Immunology community at Hopkins, as well as all the future immunologists this pandemic has made /enlisted. I have to say, it sounds like a thank you at the end of a thesis defense, right?
I like it.
Thank you, Jesus.
Yeah, that was great. And so I think if I remember back, maybe we were not as critical in regards to how the peptides were generated. But if you go back and listen to John Udell on TWiV, which anything John Udell does, you should listen to. I remember he did critique this paper. He did mention that this maybe would not even have been published in such a high level journal because of this issue about the peptide generation. And that it's very difficult to know the sensitivity of those peptides when they're generated in a pool. So I think people were thinking about this, but what we learned over time is that this evidence of cross-reactivity does exist, and we fine-tuned it over time. I think, if you were the author of this, you would maybe also weigh that in your mind. Do we want to get this out there, or do we want to take 6 months to a year to use HPLC to generate these peptide pools? So, what do you all think?
I think that there's value in different ways of addressing the same question, and maybe there are limitations to each approach, but when you put them all together, you get a lot more information. Mm-hmm. And so, yes, I think there are caveats. I think where the problem is that they didn't discuss those caveats to their study, because I think whichever way you choose to do something, as long as it's a scientifically sound approach and you acknowledge the limitations, you can still learn something, right? So perhaps this, sparked some more interest in this, and people went and looked at it in a slightly different approach and gained more information. Then you add on to that. So, should they have not done it? No. Was it perfect? No. But I think there's still value in doing things different ways. I don't know. What do you think?
Yeah, I think there's value in asking the questions and starting to address some of this information. I think that perhaps given the fact that this paper was perhaps going to be clearly read by a lot of immunologists who might not all have as much background in peptide pools, it would have been particularly nice for them to talk a little bit about those caveats. Yeah. And I'm really glad that I've learned all this information from this email and can start thinking about this more critically in the future.
I think that's probably a reviewer. That would be something a reviewer should have suggested. Yes. That they include these caveats.
Part of the problem here is this idea of high-profile journals, right? I think this is nonsense. And we've talked about this for ages on TWiV. Things should be published in a democratic fashion. And then, what's important will rise to the top, be tested and confirmed and so forth. But to have a journal identify this as high profile is just fundamentally wrong, in my opinion. And that's the problem here. This is a high profile journal, and there are issues with this. And in another era, it probably wouldn't have been published here because it's SARS-CoV-2. And that's what brings it. And that's just not fair. It's not fair that being in Cell will make it more accessible than some other journal. It's not as high profile. It's there. It's out for everyone to see. Everyone can find it. You don't have to be in. What you're doing is driving the profits of Elsevier and Nature and so forth. And I think that's unacceptable. And that's the problem. Part of the problem here, because you could say, I don't know, what's a less high profile immunology journal?
Journal of Immunology. It's a solid journal.
Journal, right?
Association journal, but it doesn't have the impact factor of a cell.
This impact factor is nonsense because then committees use that to say, oh yeah, let's hire this person. They have 5 Cell papers. Right. Without even reading the papers. That's ridiculous. And so, in my field, Journal of Virology is a solid journal. Lots of things are published there that you would not say are high profile, but it doesn't matter. The good stuff will persist in the literature. And I think we have to change this publishing model that rewards this high profile nonsense.
Yeah. And if coming at it from a trainee perspective, I could give you my thoughts on this because I agree with all of that. And I think bioRxiv has allowed some democracy in publishing to enter the fold. I will be putting my work on bioRxiv, but I also will be submitting to these high impact journals because my career could be propelled by a cell. It would be propelled by a cell nature science. And I have no control over that. I would love to take a stance and say, I will only apply to open access, non-high profile journals. But until it comes from the top down,
of course, it has to be fixed before you can. I totally get that.
Yeah.
It has to be fixed. Otherwise, yes, you should publish in these journals. Otherwise, you're going to put yourself at a disadvantage, right? But I would argue that you don't, in the end, we don't need this. The good science will be recognized, right? Oh, let's interview this person. These 5 papers are an amazing concept. It doesn't matter where they're published, right?
Right.
Yeah.
Right.
That's all
I think there's a few interesting points that I've seen floating around lately. One is the cost to publish in some of these journals. There was a whole thing about the Nature costing like $10,000 to publish a paper. That's prohibitive for most labs. The vast majority of the labs certainly cannot afford that cost associated with publishing one paper. And so that itself is an issue. And then there's a few other things. There's one thing that says like, oh, well, senior scientists should not publish in those. So you can give those spaces to junior scientists so that they can, make tenure and so forth, which is an interesting argument. And then there's the arguments by some senior scientists that we should all just stop publishing in those journals and only publish in open access journals and, more lower profile journals that are solid. Sure. And that everybody should do this, right? And it's easy to say if you're sitting in a full professor position with tenure, right? But then you're evaluating these people that you're expecting them to have made a name for themselves. And then how are you evaluating whether they made a name for themselves? We're still using the good old, check boxes.
Right. Yeah, I find that third argument to be rather frustrating. As someone with a very small lab, for whom a lot of open access fees are quite prohibitive.
Yeah.
Yep.
I agree that, yes, it would be great if we could publish everything in open access journals, but there's no way that a lab my size could afford that. Right. So I think that those are frustrating and difficult arguments.
This idea that you could get all scientists to agree not to publish is just absurd, because you would never get scientists to agree. That's the basis of science, that you're not going to agree, right?
Sure. And if I could throw one idea out there that I think could work, if there was a person who had the time and the charisma to marry kind of together advocacy around the public outrage of this. Because if you really think about it, we are taking the public's money through taxpayer dollars. We're giving them to researchers like ourselves, and then we are paying multi-million-dollar publishing companies to publish that work that they cannot access because it's closed access. Yeah. So I think a groundswell of public outcry to Congress, to whoever, that maybe would drive it. But you would need a central figure who has charisma and who can really get to the public. So unfortunately,
somebody needs to do a TED Talk on this.
You know what? It's been done by many people. The problem is Harold Varmus, former NIH director, has on record saying, NIH accepts publication fees as part of the cost of supporting a grant. He's on board with doing this. Yeah, because you would argue that maybe this should be. Maybe the NIH or some part of the government should publish all academic research, right? In a democratic way where everything is equal, and pay for the cost. And someone would say, well, then they should just give, $2,000 per paper per journal article. But anyway, this is not something easily solved.
No. And I think when you consider that, I like that idea that the NIH would just publish all work and you would have to cipher through. But I think ego, one of the things of being a scientist, why we want, we want awards. We want to win the Nobel Prize. We want to, we want to publish in Cell Nature Science. So it's hard.
Well, I don't that much. I'd rather do podcasts actually, but.
No, I know.
Most people do.
Might not feel the same way.
But David Baltimore is on record saying, a big driver of science is ego. Yeah. And he's right. I used to have that on my lab door for years. It's ego. And this idea, you can think of so many PIs who would never not publish in Cell so that a young person could do it. Never. Just you can think of them. I'm sure many names would come to your mind. Anyway, I'm sorry to get us into this nest of.
It's interesting. I think people are interested in kind of the workings of science. But thank you to Jesus for that.
That's a great email.
Letter.
Yeah.
That was great.
All right, it brings us back to Cindy.
I'm up. Okay, Jacob. Right. Good morning, TWiV gang. So maybe he sent this to TWiV, but it's better here. It is a comfortable temperature this morning in Boise, Idaho. I do not check the actual temperature when I first get up in the morning, but simply step out and decide whether I need to wear a jacket or not when I ride my bike to work. I tried to listen to TWiV 301 on viruses in the ocean, but my iPhone gave out 5 minutes into the podcast. So I switched over to number 544 since I had already downloaded that one. I had a question on immunodominance. So that's how it ended up here. Okay. So excuse me if this is a new idea for me, and my question will probably be somewhat imprecise. I understand that there is no dominant epitope in influenza viruses. Is there an immunodominant epitope in the hepatitis A virus? I assume that for hepatitis B, the immunodominant epitope is the surface antigen. Please correct me if I am wrong. The reason I ask this question is that I heard on a YouTube lecture that any antibodies to hepatitis A virus are protective. I just want to make sure I understand this idea. Do we want to answer that question first before we go to the next one?
I think this came to both. That's why it's here. I wouldn't have.
Okay. So I think that everything has an immunodominant epitope. It just matters that they might be different from antigen to antigen, because, if you break up an antigen into multiple different peptides, one of them is going to work better than another, right?
Right.
Do you agree with that? So
I would agree. Yes.
I don't know. Maybe I'm not sure what we mean by immunodominant epitope in this instance. Maybe a immunoprotective epitope, like it's very hard to get one or a cross-reactive, universally protective epitope. What is your thought on this?
I think part of the problem is that he doesn't know what an epitope is, right? Because he says the immunodominant epitope is the surface antigen. That's a huge protein that has many, many epitopes in it, right?
That's right.
So that's the first thing. An epitope is small. How many amino acids typically?
It can vary. Yeah. It varies because it can be three-dimensional structure of a few amino acids to a
So it's not the whole protein.
10.
Yeah.
And if you look at influenza HA, so when you get infected with influenza, you make antibodies to HA and many other proteins. There are some epitopes that are dominant, immunodominant. You see, and the stem epitopes are immunosubdominant. You don't see many of those, right?
Right.
But this idea that all antibodies to Hep A are protective is just not right, because there are going to be some that do not neutralize and others. So that lecture, wherever you heard it, is wrong.
Right. Yeah. I think that the idea here is that so maybe a subtle thing that we could mention is that if you have a large antigen and you can break that into small little pieces like we're talking about, or have different regions on the surface that can be bound by different antibodies, certain epitopes will be better at generating an immune response. That may work or it may not, because that epitope, which is really good at generating antibodies, may actually be buried. And so the antibody you generate is useless. Right? So it depends on, like, if you're using an epitope to immunize, you might use a epitope that's really good at generating an immune response. You get lots of antibodies, but they don't do anything because the epitope that it recognizes is buried. So we wouldn't normally do that. So when you decide on what you're going to use for immunization, you want to look at both surface exposed. So when you generate an antibody, it can bind to the surface, and also a sequence that has actually in the past been really good at generating an antibody response. So that's how we decide on, what we would guess would be an immunodominant epitope. But I guess the only way you really know that is if you survey a whole bunch of people, and a lot of people generate antibodies to one particular part of the molecule. It happens over and over and over. And so that would tell us that that's a really good epitope. And I guess we would call that an immunodominant epitope. Do you agree with that description?
So I would call something an immunodominant epitope if you make the majority of your antibodies against that antigen against that particular epitope. So just as Cindy said, you are going to have multiple epitopes within any antigen, multiple regions that your antibody can bind to, multiple short peptides. And some of them are going to induce more antibodies or more T cells than others. They're not all going to be equal. And the details of how that works are tricky and not totally understood. Right. But just because a particular epitope is immunodominant, as in you make a lot of antibodies to that one, you make the most antibodies to that one, it doesn't mean that those antibodies are going to be particularly useful. Mm-hmm. So just because it's an immunodominant epitope doesn't mean it's necessarily going to be protective. In fact, sometimes we have this problem where there will be an immunodominant epitope that sort of is almost directing the immune system away from the protective epitopes. And so I think that also that idea of protective versus the dominant epitope is being confused a little bit here. Yeah. And Jacob also probably wants to remember that epitopes for B cells and epitopes for T cells are different. So the immunodominant antibody or B cell epitope might be different than the immunodominant T cell epitope.
Yeah. I think a good example was one that Sally Permar in our last episode talked about was CMV. And what they had discovered was that they had used so the idea of the virus fusing to the cell, there's a pre-fusion and a post-fusion form of whatever the structural component of that virus is. And you may think, oh, we're going to give the antigen to the pre-fusion version and we're going to develop antibodies and that's going to be protective against disease, where in fact the cell presents that peptide differently in a different conformation. And that's the way the antibody needs to drive a response against that. And so that they had found that protecting against disease was against a peptide that was presented in a different way. And there's a whole structural biology using structural-based methods to determine this is like rational design of vaccines. When you hear this term rational design, and it's based on the structure of your antigen.
And in fact, for SARS-CoV-2, they use a mutation that stabilizes the spike protein in order to generate efficient antibodies against it, at least for the primary vaccines that are out there right now, as far as I understand.
Mm-hmm. Well, hopefully for Jacob, we were able to answer that first part of this question.
But Jacob actually had two things. So his second question is on cytokine storms. I have enclosed this paper on cytokine storms and wanted to get your viewpoint. I must admit that if you were to ask me what a cytokine storm is, I would be at a loss for words. Dr. Barker, I've appreciated your lectures on the immune system. I used them last year to teach myself immunology, and I did use the Kubi text to supplement the lectures. The innate immune system is super interesting. Studying your lectures put me in a much better position to read and understand the flood of information on SARS-CoV-2. I have been following the Virology and Immune podcasts religiously. It is the only way I can hope to keep up or try to keep up with a flood of information. Now, when I have time, I try to backtrack and listen to older ones. It has helped me focus on learning. Under the current circumstances, I cannot take a leisurely approach to my education. I'll just finish it and then we can go back and answer the question. I recently volunteered to be a PI for a serological study. Finally, after many years of practicing medicine, I get a chance to do some research. It has been a goal of mine for over 20 years, but due to life events, it never materialized. So I grabbed this opportunity when it appeared. Someday I hope to be able to do laboratory work, perhaps after this is finished. Thanks again for tickling my scientific curiosity, Jay. So that was really nice. It was a really good email. So coming back to this idea of cytokine storm, who wants to give a stab at a definition of cytokine storm? I think we all might have a slightly different definition.
Yeah. I mean, traditionally, I had thought of it as a hyper-inflammatory response defined by pro-inflammatory cytokines that increase after infection and persist. So this would be IL-6, IL-1 beta, TNF alpha. Mm-hmm. I think that my view of this has been changed with the past 3 to 4 months of what people are describing as a cytokine storm with SARS-CoV-2 may not actually be what we see with sepsis or what Ebola patients experience, because there's another component to cytokine storm that leads to organ failure, systemic organ failure, and has a lot to do with leaky blood vessels and a cascade of events that we might not be seeing with SARS-CoV-2, although we do see these cytokines. But there were some recent studies showing that with these other diseases, compared to SARS-CoV-2, the levels of IL-6 were high, but not as high as somebody who dies from sepsis or Ebola-induced disease. So I think we're learning that we can't just apply cytokine storm to everything.
Yeah, I think that the most important part of this to me is that I originally thought of this as sort of excess inflammatory cytokines leading to sort of pathology, and particularly the inflammatory cytokines that Steph mentioned. But the thing that I have learned as a result of SARS-CoV-2 is that there probably are more types of cytokine storm than we were originally saying. We were lumping together a lot of really different things. And so I'm not sure there's only one flavor of cytokine storm. And I think that is something that we need to better understand.
It's not surprising because here in the last year, we've looked at it in more detail than we ever did before. And so we're revealing things that may have been present in SARS-1, avian flu, MERS, But what do they want to call it now, Steph, instead of cytokine storm?
I think they want to call it, similar to what they did with the children, multi-inflammatory syndrome of adults. So MISA rather than MISC. I think they want to get away from the cytokine storm reference.
Well, I think one of the ideas is that we, to some degree, presume that it's the production of these inflammatory cytokines that then their signaling in response to those cytokines drives the pathology.
Right.
And I don't know that that's necessarily true. I think it's an indicator of, but certainly I've been thinking and reading a lot about the inflammasome and production of IL-1, which we can get into in detail or whatever, but that is also directly connected to the coagulation cascade.
Right,
and it's been shown much more, a lot of recent papers have connected that with these cytosolic molecules, STING and GasDRMD and whatever. It's basically that when you're producing particularly IL-1, not the other ones, but particularly IL-1, you're also driving this coagulation cascade as well. And so the IL-1 production probably is more of a biomarker in some ways of these other events that might be going on that are leading to that. So yes, some of it is if you make lots of TNF or you induce IL-6, or inject IL-6, you're going to induce these inflammatory pathologies if you have them at high levels. But if you knock them out, you don't necessarily have that pathologies. And so it's both a contributor and a biomarker and a result and something in parallel. So it's just too muddy. And I do like the idea of saying that there's more than one kind of cytokine storm, because clearly with some of these viral infections, interferon plays a huge role as well in what's going on.
Yeah. And it is relation to treatment, because we talked a lot about tocilizumab, which is an IL-6 blocker. Cindy, what do you think that means now that we're redefining cytokine storm? How does that have implications for treatment?
Well, I think you can still try to block some of these things because, you're teetering on, I at least this is my personal thoughts on this. Like, right, you ramp up this inflammatory cytokine and all these other things that are going on. And once you reach this tipping point and you're just kind of like out of control, it's very hard to shut it down. And so if you can try to block some of the effects of some of these, you probably let it simmer low enough that the immunoregulatory events can control it and you don't go wacky. So I think there's probably usefulness, but the idea that we're looking for the cytokine that goes up the most and then treat that with a blocker for that particular cytokine, and that's going to make it better, probably is somewhat myopic in thinking.
For sure.
And that's reflected in the failure of TOCI to have much effect. TOCI on its own, right? Monoclonal.
That's right.
Brianne, can you take Lindsay?
Sure. Lindsay writes, "Hi, Immune Crew. I'm taking an anatomy and physiology class, and this question came up when we were discussing blood typing and transfusions. It makes sense that someone who is Rh negative will only produce anti-Rh antibodies if they are exposed to the antigen. But it makes less sense to me that someone with type O blood will produce anti-A and anti-B antibodies without being exposed to either antigen. The sources I've looked at don't say how this happens, only that it does. How do those antibodies develop? Is there another source of these antigens besides erythrocytes? If there is, wouldn't it have to be a transient source or risk constant agglutination? Thanks for taking the time to make this podcast. The TWiVerse has been a great source of information over the past few months. Stay healthy, Lindsay. This is a great question, Lindsay. And I really like this question because it shows that you're thinking carefully about immunology, and the fact that in order to have in that type O person appreciable levels of anti-A and anti-B antibodies, you need to see the antigen to sort of prime up that response. The antigens that you're talking about here with type O, type A, and type B blood are carbohydrate antigens. The difference between type A and type B is actually in the terminal carbohydrate on a chain of sugars. And many of those same sugars are present on microbes that are in the microbiome. And so those will be the source of the antigen that can prime this antibody production. And so we all originally or theoretically would make all of these types of antibodies. But in, say, a person who has type B blood, the B cell making the anti-type B antigen is deleted by negative selection so that we don't have an autoreactive response, while the rest of them are generated when we generate those diverse B cells and then primed to make lots of antibodies with the encounters with those carbohydrates on the microbes.
Right. And so that's why, when we think about the development of the microbiome and those bacteria that contain those carbohydrates on their surface, in addition to some foods are also going to contain those carbohydrates. So it's both food and the microbiome. This is why you're not going to serum test for anti-A or anti-B antibodies in newborns, because they are not going to have those because the microbiome is developing. So that is kind of evident as to what's causing this, what's driving this. It's the development of the microbiome over time.
I hadn't thought about that with blood typing newborns.
Yeah. And it's interesting too, because this is I think we're talking about when we think about antibody responses, this is primarily an IgM-based response. IgM kind of percolates at low levels. It's not particularly, it doesn't bind particularly strongly to antigens, but binds broadly because there are 10 different, if an antibody, I'm putting my hands up like it's a Y, right? Okay, there's 2 places that the antibody can bind to, but IgM has 5 of those, so 10 opportunities to bind. So it doesn't need a lot of IgM. It can bind with low specificity to multiple different things, and that's the type of antibody response we're talking about when we talk about blood types and autoantibodies against that.
Steph, you're next.
Yeah, and actually, so we did answer that question, but I guess in regards to COVID and other viral infections. So we know with rotaviruses use sialic acid to bind to its receptor, because these carbohydrates are not just on the surface of like blood cells, they're on the surface of many types of cells. So it's not surprising. I think that was like a big thing. Oh, if you're this type of blood antigen, you're better protected against SARS-CoV-2. It's not uncommon that these things interact with infections and you may be more or less susceptible depending on how that virus enters your body and what receptor it uses. So, okay, next one. And I'm on Lauren, is that correct?
Yes, it's Lauren. Yes, correct.
Lauren.
Yes.
Okay, great. Dear Team Immune, thank you for all of your work to help your listeners understand the immunology and virology of SARS-CoV-2. I discovered your podcast in early April and I'm greatly appreciative of your insights on this virus. My question is on the detectable immune response I've been exposed to COVID-19 repeatedly. I was in northern Italy at the end of February and in NYC the first 2 weeks of March, during which I had dinner with one friend who developed a fever 2 days later and kissed someone who developed a fever 4 days later, both of whom later tested positive for COVID antibodies. I then flew to California, and 10 days later, my roommate developed COVID symptoms and later tested positive for antibodies. During the week she was sick with a 101 fever and bad cough, we still ate together, dinner together, and I was around her while she was coughing, and we shared the same space. I never developed symptoms, and as despite my friends accusing me of being patient zero, have twice tested negative for antibodies. What is your hypothesis on why that is? So let's start there, because there's a little bit else to this letter. It's good in general for all infections that if you are symptomatic or you are potentially noticing that your friends, when they're around you, are developing symptoms, that you may want to quarantine yourself. This is not specific to SARS-CoV-2. This is just protection of infection. So my hypothesis is that you could have been asymptomatically infected by SARS-CoV-2 and you were spreading it to your friends who developed symptomatic infection. We know from SARS-CoV-2 that the ability of this virus to transmit in a person who never shows symptoms is the driver of transmission, and it may be unique to this virus. Maybe not something we were fully aware of in February when this was happening to you, but would you guys agree that that would be the hypothesis?
Yes.
Yes.
Okay. Now she says she's twice tested negative for antibodies. That's curious. I think there's a couple of things there. I think the tests you take, maybe a third antibody test. I don't know what tests you took when you took them. The sensitivity of these tests have improved over time.
Yeah.
If you took those tests and when you said you were infected, take them again. I think the sensitivity is better. Next, if you did not have, we know that the correlation between high IgG levels and severe infection has been demonstrated in the literature. So it's to say that if you were asymptomatically infected, maybe you don't have levels that are detectable from a rapid antibody test. You may need an ELISA test or something more sensitive. There's also the third option that you were not transmitting SARS-CoV-2 because it's a suspected. Did you ever get a virus test? This could have been something else.
Yeah. I think the important thing is without a virus test, we don't know for sure, right? Because you can develop antibodies and then they can go back down and you might be below the detection limit of the test, or you may not have developed a large antibody response. We're seeing that repeatedly in multiple people. So, there's also lots of reasons why a test might test negative, even if you are having antibodies, for example, if it's an antibody test. Not all tests are foolproof. If you've tested twice and got negative twice, that's probably less likely the answer. You improve your positive test rate when you test twice, and you say if either one of those was positive, then we'll call it positive. So you got two chances. But I think that the point is well taken, that the sensitivity of the antibody tests a while back was not quite that, wasn't up to par, and so they're improving. Maybe if she took a test now, she would be positive. I don't know.
Yeah, because in the timing of that too, we don't know if she took it too soon, if it needed time.
I like your suggestion, Steph, that she may have had some other infection.
The flu is still a thing.
People are assuming, and we don't know, and that would certainly give you negative serology for SARS-CoV-2, right?
Correct. Yeah.
There are many viruses out there that cause these non-specific symptoms. So I just want to correct Lauren. She writes, I've been exposed to COVID-19 repeatedly. Well, that's not correct. You either have been exposed to patients that have COVID-19 or you've been exposed to SARS-CoV-2. But actually, in either case, you don't know that, right?
Right.
I mean, how do you know you were exposed to a patient with COVID-19? Do you have a serology or PCR test that said they were positive? If you were in Italy, northern Italy, you might have avoided. So don't assume that you were exposed to anyone unless you know.
Right.
Sorry.
So, no, that's good. I think that because I think she's making the connection. Northern Italy and NYC were two places where cases were booming. But that's just anecdotal, right? We need to have a test.
I was in New York City all year, and I never got infected.
Right. So the rest of this, questions asked, could my innate immune system have fought off any virus I was exposed to? If I mounted an adaptive immune response, could my neutralizing antibody levels not be over the threshold for a positive serology test? So I think we answered that question. Going back, could my innate immune system have fought off any virus I was exposed to? We could say that a viral infection, that the innate immune response contributes to protection. I don't think we fully understand that quite yet. My innate immune people, I'll let you answer that.
I think that it is theoretically possible that such a thing could have happened, but I'm not sure we have a way we could actually measure that or tell Lauren for sure.
Right. Would you speculate that I may have helper T cells that may recognize the virus? I acknowledge that without data, it's impossible to know, but I'd still be interested in your speculation. So, it is possible that you have positive SARS-CoV-2 positive T cells after you were asymptomatically infected. We are seeing that in the literature. Again, it's not to say though that it's at low levels and maybe they're mostly harbored in your organs and not circulating. So hard to know without data. And if you know any researchers in the San Francisco Bay Area looking for COVID study participants that fit my description, I'd be happy to volunteer. Thanks so much, Lauren. I know there were studies, serology studies. I'm not familiar with them now, but I'm sure, you could just try to get that antibody test again if you're really curious.
Daniel Griffin has mentioned a website where you can go and look locally to see if there are any such trials going on. I don't remember what it is, but it's probably linked to in one of Daniel's TWiVs. Yeah, we'll do one more. Jay writes, I saw this paper on UCSF Grand Rounds last week on impaired type 1 interferon activity. The presenter spent 10 seconds on the paper. That was a bit of an annoyance. Yeah, I'm sorry. 10 seconds is useless, right?
It's a great paper, too.
Yeah.
I had several questions and thought it might be a good discussion for everyone. The paper on page 2 says that plasmacytoid dendritic cells are the main source of interferon alpha. I think my problem is I do not understand how dendritic cells are classified. The paper appears to suggest that alpha and beta type 1 interferons are affected differently by SARS-CoV-2. I'm perplexed. What are plasmacytoid dendritic cells good for? Kubi 8th edition, page 95, says that they secrete type 3 interferons. All right, let's stop and take that. Who wants to tackle that?
Go ahead. I think this is more your wheelhouse, but I could comment at the end.
Okay. So dendritic cells,
are awesome.
They're awesome. And as I always say, immunologists like to subclassifications of subclassified cells. So there are dendritic cells, which are your traditional, what we, think of as the standard textbook antigen presenting, cells, professional antigen presenting cells. Plasmacytoid dendritic cells can do that, but we usually call them the type 1 interferon producers. So they're really good at taking up and cross-presenting antigens, and we've talked about cross-presentation before, but they also produce high levels of type 1 interferons. Kubi is saying type 3 interferons, they do produce those too. So what we typically tend to do if we're not interferon immunologists is lump type 1 and type 3 together, because we say they do the same things. I'm doing air quotes here. So I think that now that we can do single cell sequencing, we are discovering that interferons are not all created equally. And so we've always thought of type 1 interferons, interferon alpha and beta, bind the same receptors and do all the same things. But that's probably not entirely true. Why would we have lots of different ones if they all did exactly the same thing, right? So I think we're now discovering that there's multiple different ones. But there's also not just alpha and beta. There's omega and all these other ones as well that everybody kind of shoves under the rug and ignores. But they may actually play a really important role in certain diseases or certain viral infections that we don't understand. What's really important is that type 1 interferons and type 3 interferons are very different from type 2 interferons, which is really unfortunate because many people get them confused. And type 2 interferon is interferon gamma. And that does completely different thing. That's really good at activating macrophages to kill things inside them and other things as well. But so the type 1 and type 3 are really good at blocking viral infections. So the plasmacytoid dendritic cells, why are they called plasmacytoid dendritic cells? They look a lot, if you put them under EM, like plasma B cell. And that's because they're almost all rough ER, because we call them basically interferon producing factories.
And ER, for those who may be coming from a non-biology background, no, it's fine. Endoplasmic reticulum.
It's where you make proteins that are secreted outside of cells. So they're synthesized on the surface of the endoplasmic reticulum to get secreted outside of cells. So I hope that is relatively clear, the fundamental differences between what we call a classic dendritic cell and a plasmacytoid dendritic cell. And this idea that there are type 1 and type 3 interferons, and we lump them all together, but they probably do different things.
Yeah, I will tell Jay, again, sad confession, as I spend a lot of time thinking about innate immunity, that I often struggle with some of the classifications of dendritic cells too. So I wouldn't, worry too much about that. And I would say that, the plasmacytoid dendritic cells, sure, they secrete type 3 interferons, but also type 1. It's sort of the type 3 is an addition to those type 1s, not only type 3.
Yeah.
Right.
And just for those DC aficionados out there, I'm not suggesting that plasmacytoid dendritic cells, or PDCs as we often refer to them, are the only ones that respond to viral infections. There are lots of classical dendritic cells, subclasses of those, and certain ones of those are much better at cross-presentation and much better at, antiviral responses, and other ones that are not. So it gets really murky, and I didn't want to go into that. But the classical versus PDC or plasmacytoid is a pretty easy distinction that you can make.
Yeah, so what I was going to comment on, I think the recent literature, based on what Cindy was mentioning, because of single cell technology, there's so much now heterogeneity amongst this functional diversification of the plasmacytoid dendritic cells that we still don't fully understand. Is the is it from one is the ontogeny of this cell subset from is it restricted, is what I mean to say? Or are they fully differentiated and they retain plasticity in response to different challenges? Do they change depending on the infection or where they're at? Because type 3 interferons, that's mostly a mucosal epithelium-based response. So I think it's still, don't worry if we're quite sure. I think we're still defining exactly what these cell types are.
Yeah.
All right. Jay also writes, I've enclosed a link to a second paper. Longitudinal analyses reveal immunological misfiring in severe COVID-19. I sent it along in case the first was not interesting enough. This paper was much harder for me to understand. I'm still working my way through Dr. Racaniello's Virology and Dr. Barker's great immunology videos. They're pushing me into worlds not yet explored. All of you rock. You're helping me to expand my reaches. Heck, I even started listening to the Parasitology podcast. They're a nice break from COVID-19. Thanks for all you do to educate the public. Anyone want to summarize this paper?
Yeah, we should let Jay know that we did review this paper. We talked about it in great detail on Immune 34.
34, okay.
And yep, we covered the paper.
Great. Immune 34. We'll put a link to that in the show notes. Okay, that'll do it for Immune 39. We didn't really get through many of these, but nevertheless, we had nice discussions, and we hope you learned from them.
We'll do more email episodes.
We will do some more. You can find the show notes at microbe.tv/immune. You can send your questions or comments to immune@microbe.tv. If you like what we do, consider supporting us. Go over to microbe.tv/contribute. Cindy Leifer's at Cornell University on Twitter. Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langle's at Duke University. Stephanie Langle on Twitter. Thank you, Steph.
Yeah, this was fun.
And Brianne Barker's at Drew University. Bioprof Barker on Twitter. Thanks, Brianne.
Thanks. This was great.
I'm Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.